And so our first speaker today will be Dr. Duxin Sun, the Charles Walgreen Jr. Professor of Pharmacy, Professor of Pharmaceutical Sciences, and the Associate Dean of Research for the College of Pharmacy. And he'll be talking on pharmacokinetics, core, and mass spectrometry. Thank you.
Thank you. Thank you, Evan. Thank you very much, everyone, to come this early to listen to the core presentation. So pharmacokinetics core and mass spectrometry. So we just use the pharmacokinetics as a sample. So this core was set up in 2009, about 15 years ago. I and Emmett Pai serve as the co-director. I mainly focus on preclinical side, and Emmett focus on clinical side. Our managing director is Baldwin. He's here somewhere. The pharmacokinetics core have three goals, or three objectives. Number one, or we can call three aims. And aim one is to help the community to have quantitative analysis, LCMS, all the small molecule, metabolite, any small molecule, most like any small molecule metabolite you want to analyze, you want to quantify. Also, we set up mass spectral imaging. We can start doing some of the small molecule metabolite imaging on tissue section. So that's, I think, imaging seems to become a very attractive area for everybody. So that's aim one. Aim two is to help University of Michigan drug discovery community to optimize the lead compound to advance to clinical stage. So we do preclinical pharmacokinetic study, drug metabolism, soft spot identification, and pharmacokinetic in animal model. We call that to optimize drug-like property so that you can move forward. Number three, aim three, is to help clinical pharmacokinetics really help that sample analysis, also modeling, to help to optimize the dose regimen in clinical trial. So we mainly support phase 1, phase 2 clinical trial. So those are the three aims of the core. So dive in a little bit detail for each aim. Aim one is mass spectrometry imaging. So in theory, we can imaging metabolites, lipids, drug, small peptide, and small proteins. So normally, from the diagram you will see, we can get a tissue section either from human or mouse. We can use frozen section. Once you put the glass slides, then we put the matrix, the usually multi-matrix, then we directly scan that in the mass spectrometry by the molecular weight. So you can see we don't need a label. Nothing labeled. In theory, we can monitor all the molecules there, but there's a challenge. So once we get the signal, then the software will go back to reconstruct the image to see where the molecule are. Are they in cancer cell, stromal cell, blood vessel, or extracellular matrix? It's very easy to do a molecular scan. So for example, if we don't mind what a molecule there, so it's very easy to see. I would scan all the molecules from 1 Dalton difference. I can scan all of them. I can construct an image. So that's very easy to do. So that's actually, everybody can get that image. And not very useful until today, because you can have a very beautiful pattern. And especially brain, you can get a beautiful multicolor brain image. All the molecular weight structure there, for sure they'll be different. They were different in different region. They were different in disease area. They were different in cancer was normal, for sure. But the problem is, what are they? That's harder. And until today, not very useful, unless in the future, I envision we can scan every single tissue, more like HE staining. Assuming you can do HE staining all of them, then we can scan every single image. You have then every single tissue with all the molecular weight, you can do a pattern recognition. If everybody uses machine learning algorithm, they can usually start to distinguish tumor what's normal. But so far, I think it's too much work. I don't think people accumulate enough image yet to do that. I can actually imagine in the future that's maybe one of the tool very similar to HE staining, but you have a molecular information and have a much higher resolution. It's harder to image all the known compounds. For this example, you can imaging all the phosphotype lipids and in different regions of the brain. And you can also image a metabolite. Those are the lipid metabolite, the adenosine, glucanate, glucose, or other TCA cycle. And people already published that, small molecules, and a peptide, and some of the proteins. So we did for one of the company, they wanted to know where the location of the small molecule in voclosporin, cyclosporin, tacrolimus. And especially cyclosporin and voclosporin, only difference is 2 carbon difference. The structure is extremely similar, but only 2 carbon differences. Then you would not assume, you would not expect there will be much difference in terms of everything. So, however, cyclosporin is approved for transplantation. The #1 side effect is kidney nephrotoxicity. Ironically, voclosporin is very similar, is approved for lupus-induced nephritis. So that's actually counterintuitive. So if this class of drug induce nephrotoxicity, why you can use this drug to cure to treat nephritis? So they really try to understand that. So by imaging, we can see this drug distribute all the matrix cortex, perhaps in the nephron, and this one rapidly cleaved from kidney, they went to urine. You can see actually later, as we found, this drug actually, you can find a much higher level in kidney, and this is lower. Then you would worry they will cause kidney toxicity, but they don't. Why? Because they quickly excrete it. They don't stay in the tissue very well. So that's we help the company to did the imaging study. They are really happy to understand why this drug is so unique in terms of efficacy and toxicity. So another thing we do is we are just able to start to set up the immune profiling using imaging. Then this one we still use antibody. We use antibody to identify each type of immune cell, CD4, CD8, macrophage, or B cell. Then the antibody are tagged with the tag. Once the laser shine the laser onto the tissue, then the tag will be released. We can detect the tag. So we are able to detect the immune profile in the tissue. The hope is you have immune profile immune cell, then you can have another layer, another tissue section, or another layer of small molecule, then you can overlay them together. So we are working on that to see how that go. Small molecule, you have much more resolution, but so antibody, the resolution is not as good as fluorescence. But the advantage is you can really multiplex small molecule versus larger molecule, because most of the time for small molecule, we don't have antibody for the immunostaining. So that's for imaging, because I've emphasized a little bit more since this is very imaging oriented. Quickly, I go through the aim #2. We help the community to optimize the lead compound in drug discovery. So far, we are able to, we have done assay for about 8,000 compounds, all small molecule. We are able to help microstability, metabolite identification, protein binding, all the drug-like property we're able to optimize for the community. And also we do animal model with dose, with sampling, we analyze, we do some of the PK modeling. We also do multiple tolerance dose, not toxicity per se, but then to see how high dose can go. So the overarching goal for the preclinical lead optimization is really to help to improve the drug discovery successful rate. So currently, when we do all the drug discovery, the drug discovery successful rate is about 10%. In cancer, is about 5%. So any compound went to phase 1 trial, 1 out of 10 will succeed, 9 out of 10 will fail. That's only if your compound went to clinical trial, phase 1. Actually, to anybody, if you have a compound pushed to phase 1 trial, that's a big deal, right? How many phase 1 trials University of Michigan ever had? Not very many. Even you reach there, 90%, 95% will fail. So then it's really, we needed to help to avoid that situation. So the current thinking is the reason you fail all those clinical trials because either you have a disease target not validated, or you have a poor translation from animal to human, or genetic epigenetic change, or heterogeneity among primary metastatic patient, or different patient population, or just simply poor clinical trial design. Those only valid if you have a good drug. If I give you a poor drug, they're not supposed to work, then you can do all of those will not give you the answer. So I think the problem today is a lot of a bad drug not supposed to go into trial went to trial. Then the physicians spend so much time just to try to rescue that bad drug. So we try to help is, I think today's drug discovery for many, even industry, when we try to optimize the potency specificity, we mainly focus on the test tube, which all those IC50 in terms of potency specificity are not clinical, not those relevant, then we waste a lot of time. Second, we really don't know the drug molecule went to tissue, went to cancer, or went to immune macrophage or immune cell they're supposed to go. We really have no idea. So really, we believe today, most of the effort focus on here, then you will never reach a successful rate. Ideally, we really want a drug going to cancer, going to macrophage if you immune oncology drug, then at the same time, they have a high potency specificity. We really should focus on that, but the whole field focus on here a lot. That's where the failure occurs. So that's for number 2, aim number 2 for preclinical DMP case study. For aim number 3, we help the clinical trial optimize the dose. We help the sampling, we help analyze the sample, analyze drug concentration. We do some modeling to see what are the best dose you should move forward. The reason is in terms of dose optimization, we learned from animal model what the PK profile look like. We know the efficacy, toxicity. Then we learn from here, we move to human. Once we move to human, then we will confirm different dose regimen. Then once we learn, we'll apply to patient population. Then population could be and big and small, old and young. So that's also need to optimize. So the Amit, another co-director, focus on this area a lot. The reason we really want to optimize the dose regimen is because when you come to phase 3 trial, you have a chance, you have one shot, you have one dose or maximum 2 doses. If you fail, you fail, that's $400 million stop. That's a $400 million gone. And nobody say, like in animal model, say, okay, if a 5 milligram cake does not work, let's try another 10 mice, 15 milligram per cake. In phase 3 trial, you don't have that choice. You have one shot. $400 million. So if you get it wrong, nobody's going to go back to do it. So the problem is for cancer, MTD still used. So MTD has been old paradigm for chemotherapy. We push the dose as high as possible. As long as the patient can survive, we are fine. However, in target therapy, that's no longer true. For all the targeted therapy, antibody therapy, we should not use MTD. However, today, our third trial still use MTD. MTD, the problem, you have a toxicity, then patient will discontinue. They cannot tolerate. You actually mess up the trial. So really, from the last year or so, FDA issued two guidance. One is for oncology, you really need to optimize the dose. The MTD no longer apply, and they actually issued two guidance on that, and they have a paper on that. So dose is one of the problems. Second problem is I actually did not realize, I don't know if you guys realize that, only 30% approved, so 5% drug successful, but only 30% of 5% able to extend patient survival by 2.5 months. I actually don't know that. And the drug is approved through PFS, progression-free survival, all more than 2.5 months. Then phase 3 trial confirmation, only 30% able to extend 2.5 months. 30% do not extend overall survival even for a day. Most drugs still on the market. And 40% in the middle, extend up 2 weeks, a month. So that's the problem. One is because of the dose, another one is the wrong drug went to clinical trial. So we try to help on that, and that's the equipment. We are very well equipped. We have 8 LC-MS to have imaging capability. Let's finish. I stop here, and we have a 5 minutes question. Yes, thank you. 5 minutes question. Thank you. Yes. You mean the dose? You mean IVC for the dose is extrapolation? Yes. So regular agency, when we do first human trial, they use allometric scaling. They have a very clear guidance, but that guidance is really to make sure you don't kill a person. So they found the toxicity dose in animal, then they convert that dose to human called NOAEL dose. That's the dose will kill, they cause toxicity. Then they apply a safety factor by 50 to reduce 50-fold to make sure your first starting dose is okay, not accidentally kill the patient. So that one's still in use. However, that's 50 times lower than you're supposed to do, right? Then you can do 10 different doses or 20 different doses and treat your right dose. Then those people don't have time and money, then they have to use another scaling. So that's still used, but the other IVC actually predict a clinical dose is not very good. Yes. So we have more experience in small molecule in terms of analysis. In terms of PK, they are similar. So in terms of modeling data, then we can help. But in terms of data, in terms of get the sample, analyze the PK, that's pretty much our ELISA. We actually don't do a whole lot. So we focus on mainly small molecule. Yes. The imaging. The imaging, so after we done that, the image is burned by laser. So really, we may able to do another scan. So really, would be our part would be the last part. Oh, we use the serial section. So after serial section, then the tissue is destroyed. Thank you.